All right, so everybody, welcome to Darien Lozon's MS thesis defense. So Darien came to us back in 2018 from Cornerstone University, and she got her Bachelor's of Science degree there in environmental biology. And so been here 3 years, which is a little bit long for a master's, but no fault of her own. Brought her on for a mitigation study and best laid plans in terms of mitigation and whatnot. I think we started that project in maybe 2015 and we're in 2021, and we still have not done actually any mitigation on site, but she was able to do some pre-survey data, so we have that as part of the thesis. And then I had asked her to research some other potential topics and figure out what else she could do in the watershed. And we talked about different things. I think we started out with snakes and decided that they really weren't, had to be pretty difficult study to do. And so we ended up developing a couple of chapters around turtles. So anyway, today Darien will be presenting her thesis on wetland mitigation as well as on turtle ecology. So Darien, I'm going to hand it off to you.
All right, fantastic. Thank you so much, Jim, for the introduction. And thank you so much to everybody who came to my thesis defense. I'm super excited, and it's a great privilege to discuss the research that I've been conducting since 2018 about the ecology of freshwater turtles and other wetland wildlife in north central West Virginia. And just to get an idea of where everybody is coming from geographically, if you could just put in the chat, I won't be able to see it until later, but just to give an idea of where everybody's coming from, go ahead and toss in the chat which state you guys are coming from. I'm really interested to see where all you guys are at. But specifically with West Virginia, we are in a really unique spot in the central Appalachian Mountains, where the entire state is encompassed by this really unique system. And I will never forget the time in spatial hydrology class where Dr. Mike Strager taught us that if you were to iron out the entire state of West Virginia, the surface area would be greater than the state of Texas, which is nuts. And that's primarily due to the multiform topography that we have in West Virginia, as well as the elevation gradients that we have. And as Jim mentioned, over the last 3 years, I have had the opportunity to learn more about the Upper Deckers Creek watershed, which is located in north central West Virginia, specifically in Preston County. And this particular watershed is a mixed use watershed, similar to a lot of other watersheds within the state of West Virginia, where it is primarily hardwood forest habitat that has been fragmented over the course of time with the presence of agriculture, as well as various degrees of development and roads. And specifically in West Virginia, we have a lot of coal mining influence, and this watershed is no exception. And along the lines with my project, I surveyed 39 sites within the Upper Deckers Creek watershed. And this area, to give you an idea, is around 7,778 hectares. And within that area, I surveyed 29 sites in 2019 and an additional 10 sites in 2020, where I surveyed turtles and got various data regarding landscape and habitat characteristics. However, before I get into the methods and results of those chapters, I want to emphasize the J.W. Ruby Research Farm, which I will probably be referring to throughout the course of this presentation as Reedsville Farm. And at this particular area in the Reedsville Farm, as Jim mentioned as well, between 2016 to 2020, I have been conducting pre-restoration monitoring of biodiversity of various taxa within this wetland watershed here. And the primary reason why we need to monitor these wetlands is because of the historical wetland loss that the United States has experienced over the past 50 or so years. And that is primarily due to the fact that developing on wetlands is very difficult. It doesn't form that foundation that you need for constructing buildings as well as roads and other sorts of infrastructure like that. And wetlands have been seen to not be very productive. So whether it be row crops or hay and pasture for agriculture, or for timber and logging for silviculture, these wetlands have been converted to be more productive in those contexts. Specifically in West Virginia, we have lost locally the natural wetlands of 24% that has been found in the state. In order to account for this loss of wetlands in the state and nationally, the US Department of Agriculture, the US Army Corps of Engineers, and the US Environmental Protection Agency has implemented this boots on the ground mitigation effort. And this dichotomous key is a very gross simplification of the process. If any regulators were in this call, they would probably be chuckling, because each of these circles or each of these determining yes or no is probably about a 10 to 15 page report that they need to submit when looking at a project. But ultimately, what we want to know is, if we're conducting a project, is it near a wetland? And if it is, are we able to avoid possible impacts on that wetland? And if the wetland is impacted, we need to rectify that impact by making sure that there is a balance of quantity of wetlands within the area. And this is kind of where the compensatory mitigation comes into play here. Later on, the US Army Corps of Engineers and the US EPA implemented this idea of monitoring not only the quantity of wetlands, making sure that balance was there, but also making sure that the quality of wetlands was staying intact, because different wetlands provide different ecosystem services. And that is where the pre-restoration monitoring and my research at the Reedsville Farm comes into play. In 1969, a headwater reservoir was implemented primarily for the reason of providing drinking water for nearby towns, as well as providing a source for floodwaters to decrease the amount of flooding that occurs in nearby residential areas. And in 2011, the US Department of Agriculture Natural Resources Conservation Service deemed the dam as a hazard, and it did not fulfill performance and safety standards. So in 2014, there was a plan proposed to the USDA and RCS to make sure that the impoundment was being increased, so it was able to account for that stormwater and other things like that. And that's kind of where the Reedsville Farm mitigation plan comes into play, because that natural stream would be lost to the increasing of that impoundment. And cover treatments were applied to the area. They haven't quite been implemented yet, so all of the data that I'll be presenting today will be pre-restoration data. And ultimately, our goal was to establish a baseline of relative abundance and diversity for multiple taxa, which are different groups of animals, whether they were frogs and toads, which are collectively known as anurans, benthic macroinvertebrates, which are small insect-like critters that are either in the wetland substrate or the stream substrate, as well as birds, fish, plants, small mammals, and turtles within the riparian wetland, getting an overarching, all-encompassing picture of what this wetland is like before those monitoring cover treatments are applied. And because we covered so many taxa, we had a lot of different monitoring efforts and frequencies to account for different life histories of these animals. For instance, with frogs and toads, because they have different times in which they're singing because of their different breeding seasons, we accounted for that variation by surveying between late February, which is around when frogs start singing in the early springtime, to around early June, when the singing starts to wane off and the frogs are no longer breeding. Along the same lines, or alternatively, birds were surveyed year-round, once monthly, to account for not only the species that are locally there year-round, but also the migratory species that are only there for about 2 or 3 days, and then they either head north or head south. Plants were really interesting too, that we needed to take into account of, because of the different blooming seasons that happen. particularly with species that are flowering in June. We needed to survey them then, as well as in September or August, to account for the different flowering periods for different species of plants. Ultimately, with all of the surveying from 2016 to 2020, we found that plants were compared to other taxa, there were more species detected or found during those surveys. And with the dominant species for each taxon, we found that spring peepers were the most common frogs, song sparrows were the most frequent birds found, creek chubs for fish, earthworms were the most dominant macroinvertebrate taxon. It's important to note that macroinvertebrates specifically were only identified to class, or for earthworms, because they are so hard to identify, we just lumped them within their subclass Elaticida. Sallow sedge was the most common plant. Small mammals were the meadow voles, along with snapping turtles being the most common turtle. And snapping turtles were almost the sole species of turtle found. We only had one painted turtle captured in 2019. With that abundance data, we were able to calculate Shannon diversity as a mean diversity for each year surveyed. And looking at these different lines, the birds as well as soil macroinvertebrates and plants seem to be pretty consistent throughout years. However, the other taxa were pretty distinctly different. There was a lot of zigzagging and jig-jagging going on, and further investigation would need to be done to determine why those changes are occurring over the years. And the fact that turtles are not mentioned here is not a mistake. You can't really do much diversity indexing with only 2 species detected. And for plants, it's important to note too that we lumped all native and exotic plants together for this diversity index. And it would be really interesting to look further into how the natives versus natives and exotics changes. And because of those different zigzagging and jig-jagging, and some of them were consistent and some of them weren't, it's important to note that we need to keep this multi-taxa monitoring going on within wetlands, just because different wetland quality characteristics can affect one taxon more than another. For instance, if there was a water quality problem, anurans would probably be more susceptible or sensitive to that change than birds, because they're a little more inactive or they're a little more passive when it comes to using the wetland. And ultimately, like I mentioned previously, these pre-restoration data can provide that baseline for abundance and diversity once these cover treatments are applied, and we can compare to possibly identify if there are any possible problems with the mitigation or if anything needs to be modified to ensure its efficiency. And one interesting thing that I was just curious about, and I was just really surprised that there weren't many unique individuals captured of turtles. Like I mentioned, snapping turtles were almost the sole species captured, with one female painted turtle captured in the summer of 2019. And I wondered if maybe it was because it was a stream, or maybe something about the habitat quality or something along those lines, which ultimately led me to developing my second chapter, which is relating to freshwater turtle relative abundance and also movement based on these habitat characteristics. Is Ruby Run's channel too shallow or too narrow or something along those lines, trying to get into that nitty-gritty of what habitat characteristics are favorable for these species. And for the remainder of my talk, I will be discussing the painted turtle, which is Chrysemys picta on the top of the picture, and snapping turtle, also known as Chelydra serpentina, at the bottom of the picture. And obviously size is a big difference between these species, but there is also differences between how these animals are interacting in their environment because of their life history differences. For instance, with snapping turtles, these guys are more secluded, if you will. They are ambush predators, so they like to burrow in the substrate to lie in wait for their food. They also don't bask as regularly as painted turtles, while painted turtles are more active predators, and so on. And looking at these different habitat characteristics, these turtles might be using the same wetland completely differently because of that. For instance, when it comes to wetland type, for snapping turtles specifically, when they're moving throughout the year, they could use different wetland types for different purposes within the year. For instance, with a wetlands like this, found at Fairfax Pond Ray Wildlife Management Area in Arthurdale, this snapping turtles could use this as a habitat for finding mates, as well as foraging and other things like that. They could also use it for hibernation, but they could also move to hibernate for however long, however many months they need to stay for hibernation in a nearby stream. So taking into account of that, because streams are less likely to freeze over, they could be more quality hibernacula than other smaller wetlands that are more likely to freeze over. And that leads to the idea of wetland size being a factor and affecting how many turtles are maybe even using this area. Because ultimately, the idea is the larger the wetland, the more space there are for turtles to inhabit. And we also have the idea of wetland perimeter, which allows for more potential basking areas for species like painted turtles. And we also have wetland depth, which accounts for possibly different temperature gradients within the wetland, because these guys are ectotherms, which means they are getting their temperature from their environment. They need to have those different possible microhabitats that they're using. Vegetative cover, similarly, they have the potential for cover as well as good hunting ground for smaller organisms such as those macroinvertebrates that I surveyed earlier, as well as tadpoles and other organisms like that. And just using it for cover for hibernation or just for either hiding from predators or waiting for prey. Canopy cover similarly is that sort of idea for potential cover, but that also possibly affects basking, because if a wetland is completely covered by trees, it's less likely to allow that sun to penetrate through for those basking turtles. Soil composition is important for nesting primarily, because certain species such as painted turtles prefer more clayey soils for nesting, while snapping turtles prefer more sandy soils. Taking a step further from the wetlands and looking at what sort of things are affecting them either directly or indirectly surrounding these wetlands, we could look at different land use practices such as the agriculture or distance to roads or even other wetlands, like I mentioned previously, but they could potentially affect movement as well as indirectly affecting, because for instance, distance to roads not only directly affects turtles if they're moving because of potential road mortality, but there's also the idea of road noise being a disturbance or just being a pollutant, if you will, for turtles, because there are some species that have vocal cues. And there was one really cool study that was presented in the British Ecological Society conference in 2020 that looked at snapping turtle nestling communication while they're still in the nest incubating, which is super cool. I'm not at all affiliated with the research, but if you're interested, I can always share the Zoom link with you after the talk. And distance to nearest wetlands is really important too, just because we want to ensure that the meta population is sustained by allowing that movement between subpopulations to ensure genetic diversity and other factors like that. With all of this in mind, we ultimately wanted to find out what wetland and landscape level characteristics affect painted turtle and snapping turtle relative abundance. We also wanted to quantify the influence of land use practices on specifically snapping turtle movement among wetlands. And throughout the rest of the presentation, if you see a red circle in a picture, that indicates that there is indeed a snapping turtle in the picture, even if you don't see it, which is kind of fun. And for these remaining two chapters, the methods are fairly similar when it comes to turtle trapping. For each of my 29 sites in 2019 or 10 sites in 2020, we set 10 hoop net traps of various sizes within each site to account for potential size bias when it comes to capturing turtles, because larger turtles are more likely to be caught in larger traps and vice versa. but we also made sure that they were far enough away to ensure that we were accounting for the most coverage within the site. And for each of the sites, we made sure to trap them within different seasons of the year to account for turtles coming out of hibernation, accounting for the most active season for these turtles. And in 2019, we also tried to trap in the fall to account for any straggling individuals that weren't quite hibernating, but it wasn't as effective as we had hoped. We didn't catch many individuals, so we decided to not trap in the fall in 2020. Relating to how I tracked my snapping turtles, I applied radio transmitters to 42 adults across 22 sites. And the fact that we only tracked adults is important because the adults are more likely to make those larger overland movements compared to juvenile conspecifics. And we made sure to track these individuals every 2 to 5 days from April to October, which is that common active season for this particular species in West Virginia. To model relative abundance, we used what is known as catch per unit effort, which is kind of like what it sounds. It was the number of turtles captured per trap night. So it's not only accounting for the number of traps that are set out, it's taking into account of how long the traps were out for each session. So for each season in 2019, the number of trap nights was 30. So keeping that in mind, except for one site because one of the traps was missing. So it was only 27 trap nights. And with all of that in mind, we were able to look at different landscape and wetland level characteristics, similar to what I mentioned previously, such as wetland type, substrate depth and compaction, wetland depth, surface area, perimeter, and vegetative cover. And we also took into account of distance to nearest roads and percent agriculture surrounding the wetland from 3 buffer sizes, which were the 250, 500, and 1,000 meters. For snapping turtle movement, we, rather than looking at the distinct number of sites that a turtle used, we had to lump them into 2 groups because of small sample size. We either said a turtle didn't leave its resident wetland or it did. And we defined a resident wetland as the site in which a turtle was originally captured. And similar to relative abundance, we looked at surface area of the wetland as well as perimeter length. And because of this movement factor, we looked at more landscape level characteristics like distance to nearest roads as well as nearest wetlands, percent agriculture with those 3 buffer sizes I mentioned previously, and an additional percent sand and soil to account for that potential nesting quality surrounding the wetland. And yes, there is a turtle in that picture, believe it or not. Looking at CPUE, which is that relative abundance, like I mentioned previously, we found that there was a bit of a difference between seasons where the relative abundance for both species was highest in the spring. And with wetland type, it was really cool because we found that palustrine aquatic bed wetlands had the highest relative abundance for both species. And palustrine aquatic bed, you can think of a wetland that's predominantly covered by any sort of floating vegetation, such as water lilies, water shield, arrowweed, other sorts of plants like that. And that was compared to palustrine emergent wetlands, palustrine unconsolidated bottom wetlands, and riverine unconsolidated bottom. When looking at the model selection, we found for snapping turtle relative abundance that distance from roads, as well as wetland vegetative cover, substrate depth, and wetland perimeter were the most parsimonious model given the AICC values that were given. However, we want to keep in mind that there is a threshold of 2 for any sort of comparable models that can be looked at. And with that in mind, up to about wetland distance from roads and mean wetland vegetative cover were significant based on those AICC values. So looking at substrate depth and wetland perimeter, because they're significant, it might not be as strong of a predictor. On the other hand, with painted turtles, we found that percent canopy cover and wetland vegetative cover increased relative abundance for painted turtles, which was really interesting for percent canopy because you would expect the opposite. But yeah, that was a really interesting find for us. Regarding movement, we found that there wasn't a significant difference between sexes as to how many sites were used within a given time. It was on average about one additional wetland that a turtle used within the time that they were being tracked. However, it is interesting to note that the females did move up to at least twice as far as males. And regarding the model selection, we found that wetland perimeter and agriculture, that percentage within 500 meters was the most parsimonious model, with about 55% of the model weight. However, it is important to note that the delta AICC value was within the threshold of 2 for the perimeter only model, where there was a likelihood of the turtle leaving its resident wetland decreasing by 0.22% for each 1 meter increase in wetland perimeter. So with all of this in mind, it was really interesting to find that wetland vegetative cover increased relative abundance for both species. So if you're trying to manage for these species, having that vegetative cover is important, just allowing for cover as well as potential habitat for prey items and other things like that. And wetland structure had a stronger effect on site fidelity, which is whether a turtle stays within its wetland or not, than upland habitat. Even though that percent agriculture within 500 meters was within the top model, because that perimeter only model was also comparable, the strength of agriculture was a little weak. And although the distance from roads was not accounted for in the top models, it should be investigated further just because within the time that I was conducting this study, there were at least 4 locations of road mortality. So being able to find those corridors and identify them, so we're able to minimize that potential mortality of individuals that are sexually mature, especially for a species that has delayed sexual maturity, is really important. And along the lines of landscape and the potential effects on movement, as well as relative abundance, whether it's noise pollution or other factors like that, there are other possible direct or indirect influences on these freshwater turtles that could be monitored, such as heavy metal bioaccumulation, which is what my last chapter will be discussing, looking specifically at cadmium, chromium, mercury, lead, selenium, and zinc. And as we know, within a mixed-use watershed like the Upper Deckers Creek watershed, we can find many different land use practices, such as roads that help to make the landscape more connective for people, as well as agriculture and possible mining influence, which is fine because we need to be able to get from point A to point B. We need food and we need energy. However, these different land use practices are introducing these different metals. Either they are already in the environment naturally and they're just busting in concentrations from these different land use practices, or they just aren't found in the environment naturally at all, and it's only human induced. That's why they're there. And these metals in the wetland can not only affect the quality of the wetland, but also affect the plants and animals that reside in them as well. And that is no exception to turtles. Specifically because these turtles are higher up in the food web, they can pretty much eat anything that fits in their mouths if it's a snapping turtle, and other sorts of things like that. They are more susceptible to this bioaccumulation of metals over time. They can bioaccumulate these metals from the food that they eat, because if there's a surface runoff occurring and it ends up in the sediment, macroinvertebrates are eating within that sediment and it just rises up in the food web from there. There is also this idea that any female that lays eggs is actually transferring these metals from herself to her offspring, which is kind of a bummer for the offspring because they can't really do much about that. But there is also, while they are in their nest, incubating and developing, because their eggs are a semi-permeable membrane, they're also able to accumulate these metals from the soil surrounding them, which, if it reaches a certain level in their bodies, it could lead to potential birth defects and ultimately maybe even mortality. We as people also accumulate these metals in the foods we eat. Hopefully they're more trace concentrations because they can potentially lead to health problems in us. And the EPA has established a daily consumption limit of 0.0001 milligrams per kilogram. That is 1/10,000th of a milligram per kilogram that we could eat. And that is mostly in the context of fish, because that is the more common aquatic animal that we consume. On the other hand, the EPA also established a methylmercury criterion for fish and shellfish, which is really interesting that there is only a criterion for fish. There's nothing in the literature that I know of for turtles. But methylmercury in particular is really difficult to track, because mercury is naturally found in the environment. It's in the earth, it's in the atmosphere. We can't really do much about it, but it's when you till the soil, it's when you dig up the soil that the mercury becomes bioavailable, and critters like bacteria and plankton can then methylate it into this methylmercury, which is the most toxic form of mercury. And with these turtles accumulating metals and the potential of possible consumption of these turtles within particular areas in the United States, it's important to know what these turtles are accumulating, what the metal or what the limits are, and other things like that. And we now have ways in which we can monitor these concentrations of metals in the turtles without completely euthanizing the individual, which is awesome. And with these non-destructive tissue sampling methods, such as nails and blood, they are comparable to other more deleterious methods, such as liver and muscle tissue. And with all of that, because we are able to keep the turtle alive within the population, there are potentials for longer-term monitoring and possibly even sampling the same turtle over the course of multiple years, which is really neat. And with these non-destructive tissue methods, we are able to get different snapshots depending on the tissue types that we collect. For blood, it's similar with people. They typically fluctuate with the things that we eat, and it gives you about a 2-month time stamp of what they've accumulated over that time. But claws give a little bit of a more long-term picture of what these turtles have accumulated over time. And because this was a pilot study, we just wanted to see, are these metals in the environment? Are they being accumulated in turtles? And what do those concentrations look like between size classes and species? And we also wanted to determine, because of the potential for high mercury concentrations in these turtles, how do they compare to the consumption limits that the EPA has set? And along the same lines, if they're possibly affecting us, how are they possibly affecting the turtles? Looking at body condition. And at the end of the day, we wanted to see if there was a cumulative sort of relationship going on. If there's a possible relationship between land use and the turtles that are within these wetlands, or even affecting the soil, and going one step further and asking, because these turtles are directly relating or interacting with the soil, is the soil possibly affecting the concentrations that are in their bodies? For tissue analysis, from each turtle that we captured, we took 1 milliliter of blood from the caudal vein of the tail, as well as 1 to 2 millimeters from 3 to 4 hind toenails, which is important for some species because painted turtles, for instance, use their foreclaws for courtship, and we wanted to minimize the possible effects from that. All of the samples were then stored in a freezer until analysis using an inductively coupled plasma mass spectrometer, courtesy of NRCCE at WVU. Looking at more landscape factors, we took 2 soil samples from each site within 10 centimeters of the surface, making sure that that depth was fairly consistent. And similar to the tissue samples, we used the ICP-MS and made sure that samples were refrigerated prior to analysis. For land use, looking at land use within 30 meters of the wetland to get a more close-knit relationship with land use and heavy metals, we used a national land cover database and calculated that percent composition, and used percent instead of number of hectares because we wanted to ensure that it was consistent across sites, because there was a distinct size difference between 0.04 hectares and 7.45 hectares, which is pretty significantly different. For looking at body condition and how the turtles' bodies are potentially being affected by these metals, we essentially fit a best fit line using a log transformed carapace length, which is measuring the straight line length from head to tail on the shell. And we also log transformed weight. So with that best fit line, that was an estimated weight. That is what it should weigh based on its carapace length. However, if it was above the line, it was typically overweight. We obviously didn't tell them that if they were. But if they were below the line, it would be vice versa. It would be below the weight of what we expected it to be. And using those residual values, or the difference between the actual and expected weight, we were then able to determine a body condition index value. For statistical analysis, for looking at those different relationships between landscape practices as well as environmental factors, which were the concentrations of heavy metals within the soil, we used redundancy analyses, which accounts for the multivariate response variable, which is within each sample, they have 6 concentrations of those different heavy metals. And for the turtle tissues, we separated them by species, as well as tissue type and the different landscape or environmental characteristics, simply because we expected that snapping turtle concentrations would be higher and nail concentrations would be higher for species and tissue respectively. And similarly for the soil, we wanted to know which landscape factors were possibly affecting the concentrations of heavy metals found in the soil. So we used an RDA for that as well. To answer the question of, are there heavy metals within the Upper Deckers Creek watershed and are the turtles accumulating them? The answer is yes. Zinc concentrations were interestingly higher, much higher than any of the other metals, specifically in the nail tissues, which could be due to the fact that turtles typically run higher in zinc. It may be potentially due to their diet, but there aren't many answers in the literature that I know of at this time. Specifically looking at mercury concentrations because of the possible health implications for us, what I did was I plotted each detected mercury concentration as either a point or a triangle, depending on the species, and compared them to the different EPA limits, whether it's a consumption limit or the methylmercury tissue criterion limit. And all of the samples, blood or nail, exceeded that human consumption limit set by the EPA, which is that 1/10,000th of a milligram per kilogram. And of those 128 samples that I was able to have analyzed, 33 of them, only nails, because the blood concentrations tended to be fairly small, kind of closer to 0. But those 33 nail samples did exceed that methylmercury fish tissue criterion. The lower tails that you see in the chart are the estimated methylmercury concentrations, because they're estimated to be about 70 to 100% of that mercury that is detected with the ICP-MS. For body condition, we didn't find any relationships really at all, when it came to if any metals were related to body condition, and there were no significant differences between species. And what you see in the chart here are those residuals that I mentioned previously, so that difference between actual and expected weight. For RDAs, ultimately, this is how you read them out. So like I mentioned before, our response variable was a multivariate response variable. So for each sample, they had different concentrations of those different 6 heavy metals. And for the different predictors, those are the vectors. And vectors measure direction and magnitude. So if an arrow is longer, that means a higher magnitude of a correlation. So what we find with blood in painted turtles is carapace length is positively related to most heavy metals. And there aren't many strong relationships with environmental heavy metals. The R-square values that you see at the bottom are a range between 0 and 1, and you typically want them to be closer to 1, which indicates a strong model relationship that you have. And these values are very low, which means it's fairly weak. We would definitely need more data to look at that a little bit further. However, when we look at nails, the R-squared jumps 10 times to 0.34, which is better, but still not great compared to that 0 to 1 scale. And interestingly, we find that there is an opposite relationship between carapace length and nail concentrations, which would be what we expect because of those different snapshots that I mentioned previously. And there are slight correlations between nails and the soil, which is interesting. Selenium was positively correlated, as well as zinc. Snapping turtle blood, similarly, the R-squared values are very low. And we are finding that the carapace length is similarly to painted turtle blood related positively to those heavy metals. And nail RDAs, interestingly, did not improve their R-squared values like the painted turtles did. And we found that carapace length, similar to painted turtles, there was a negative relationship where larger individuals have lower concentrations of those heavy metals in their bodies. For wetland soil, we found that all of these heavy metals were detected, and there was one outlier site that had higher concentrations of lead and zinc, so it was removed from the analysis. And looking at the RDA, the adjusted R-square is still a bit low. However, what I found interesting here was the higher the percentage of agriculture within 30 meters decreased almost all of the heavy metals. There was a negative relationship, which would be opposite of what you would expect. However, there could be a type of phytoremediation happening where these plants are taking up the metals and they're being removed from the system entirely. So it might actually be improving the wetlands soil in some form or fashion in that kind of sense. All right, with all of that said, we want to quick look at our objectives to make sure we covered everything. And the first question was, are the metals in the wetland turtles? And the answer was yes. Were there any relationships between these species and size classes? We found that, as expected, the metal concentrations were higher in the nails than the blood, and the concentrations differed between the metals. So some would be higher, some would be lower, and so on. And carapace length was positively correlated with blood, whereas it was negatively correlated with nails in both species, which was really interesting. How are the mercury levels comparing to the EPA standard? They all exceeded the consumption limits, which is possibly a problem here. And then the 26% of the tissue samples that were analyzed had exceeded that methylmercury tissue criterion as well. We also found that the bioaccumulation of metals did not have a strong effect on the BCI across species. And bringing back that triangle again, seeing how things are related, we did find that there was a negative correlation with tissue selenium with the distance from mineland and percent agriculture. So with that idea, we have wetlands that are farther away from mineland have less concentrations of selenium. So that's possible that mineland is affiliated with selenium. We also found that percent agriculture was negatively correlated with all of the metals in soil. And we found that distance from mineland was negatively correlated with not only soil selenium, but chromium as well. And then selenium, zinc, and lead were really the only heavy metals in the soil that were related to the tissue in turtles, where there was either a positive correlation with selenium and zinc or a negative correlation with lead. With all of this said, we can use this information for possibly developing criteria for ideal wetland locations. Going back to the compensatory mitigation idea, where can we possibly create these new wetlands to account for the possible loss of wetlands to minimize the potential impacts of surrounding landscape? We could also look a little bit further at the mining history within the Upper Deckers Creek watershed and see potentially with that selenium and chromium and cadmium coming up over and over again with those possible correlations with the mine land, seeing if a wetland is remediated over time, what are those remediation factors or effects over time? And because of the mercury levels being so high, we want to see if maybe developing consumption advisories would be important for ensuring human health, especially for those within the watershed who choose to harvest turtles for the purpose of consumption. Okay, you would think that presenting all of this data would be the hardest part, but you know, I've been trying to practice acknowledgements because it's just so crazy thinking that this entire project happened over the last 3 years and I couldn't have done it without everybody. I do have to thank my funding sources as well as the West Virginia University for logistics support as well as funding. And I have, holy cow, so many people to thank. It literally takes a village to create a master's thesis, and I have at least 80 names that I have to list, which would be my Grammys thank you if I had to write one, but this is just a condensed list. I really have to thank the Society of Wetland Scientists for student grant funding and just allowing me to be a member of their society. Dr. David Samuel and his wife, Kathy, for supporting us in so many different ways. The NRCCE for analyzing all of our samples and answering all of my questions about heavy metal analysis, because I've probably bugged them so many times, it's crazy. But also at the end of the day, this project could not have happened without my interns, volunteers, landowners, lab mates, everybody. So with all of that said, also have to thank my family and my fiance and my dog and my committee, everybody, really, you guys are great. So with all of that said, I know I have more questions than answers at some point. I know you guys have questions, so I will gladly open the floor to any of that. Thank you so much for your attention.
All right, thank you, Darien. That was great. So we will take some questions from the audience. And so if you have questions, please do ask. And we'll spend however long that takes, and then we'll take a little break, and then the committee will continue to meet with Darien.
And if you're afraid to ask questions, which I know it's pretty intimidating, you could always throw them in the chat, or you can unmute yourself real quick and ask them as well, whichever is easiest for you.
There's a question in the chat, Darien.
Yeah, thanks, Krista. So the interesting thing about selenium is it is important for our bodies. Like, some people take selenium supplements if they need it. And selenium, when people look at selenium, a lot of the studies were done in the lab. So the idea of doing a project like this in the lab is you feed an animal particular concentrations of a metal and you observe the possible effects of that. Selenium toxicosis is a thing, but I don't know if there has been a certain threshold set in place yet. I think there's still a lot of fuzziness involved there as to what concentration of selenium will lead to 50% mortality in a population, for instance. So, good question, thanks. Hopefully that answered your question.
I have a question.
Okay.
Nice talk, by the way.
Thanks.
I was wondering if you explored any other metrics of turtle health other than, kind of the turtle BMI, the what you call it, the body condition index, because, for example, like mercury in humans has influence on reproductive health. And so, did you explore anything other than the BCI?
Unfortunately, no, we didn't look at any nesting characteristics or anything like that. I know a lot of studies have looked at those concentrations of metals that are being transferred from the parents. So looking at those eggs and looking at a subset of eggs and seeing what those levels are like there, but nothing like that was in the scope of my study.
Cool, thank you.
Good question.
Darien, I have a question.
Yeah, Sarah.
I was just wondering about the agriculture surrounding your wetlands. So, was it primarily crops? Were there a lot of livestock? And do you think that the type of farming would play into the types of bioaccumulation you saw?
So to answer your first question, it was primarily hay and pasture. There wasn't much cropland. And to answer your second question, yes, I do believe if we had, because the way that the National Land Cover Database worked was I believe they lumped hay and pasture together as one land use type. So because of that, we weren't able to really parse out those differences there between the different agricultural land use practices. But I would think that because you would expect cropland to increase those potential heavy metals, that maybe hay or pasture would have lower concentrations. And maybe that was why we found that negative relationship in the soil.
Well, thank you.
Good question.
Darien, I've got a question.
Yep.
So after it's been 2 years of collecting data and doing all this field work, is there anything that you would have done differently?
Differently regarding what exactly?
I guess methods wise, just overall projects. It's like things you could have told yourself back in 2018.
I should write a book. I have a list of things. So I really think ultimately the low sample size was the big issue, if you will, in this study, just because those relationships were so weak. Like I showed with those R squares are very low. They're kind of pathetic. Sorry study, but so I think if we were to maybe shift our efforts to maybe a subset of sites, saying, if there's one particular site, or maybe even like 10 instead of 22, we could say, get a larger sample size of those tissues and say, within these 10 sites, here's what we found. So just kind of shifting that, I guess, emphasis, if you will, instead of spreading out and getting lower sample sizes, we could maybe do that. So that's the first thing I think of. That's really the primary thing, I guess.
Hey, Darien, it's Joel.
Hi, Joel.
I guess my first thing is more of I just wanted a reminder and maybe a question. So with the heavy metal concentrations, it sounded like the general trend was that it seems like size is a big influence on whether there's a higher concentration in blood or nails. Is that correct between the two species?
Yeah. So because of that different snapshot, there were those opposite relationships where they were higher in blood, but lower in nails with that carapace length.
Okay.
So
was it possible at all to try or did anyone try to like remove that effect at all to see any of the underlying kind of variables that you were more interested in? Because it sounds like size ended up being kind of a nuisance variable in this case.
Yeah, it could have been, and we definitely could, just because of that size factor being something that we couldn't really change. We wanted to see that, and I kind of wanted to put carapace length in both just to see hopefully those relationships wouldn't change, because they shouldn't, between those different heavy metals. But looking at that, yeah, I don't know if carapace length would have changed those relationships, either landscape or environmental, at all.
Okay. And then I guess my second question is, since you have these ideas about how these heavy metals are bioaccumulating in these turtles, and your primary metric for health was the BCI, I'm wondering, do you feel like, based off of your results, that maybe another study is warranted to look at potentially how behaviors are changing? And maybe behaviors are how these turtles are adapting to these metals rather than necessarily BCI?
That's interesting, because I don't know if in my literature search if I've seen any studies where they've observed any behavioral changes, but I'm not entirely sure. That is a good question. I don't think that there's any sort of avoidance, if that's what you mean, like, or that sort of behavioral change. Like, I don't think turtles really, I mean, obviously that's anthropomorphizing the turtles, but I don't think they mind. Like, they obviously aren't aware, and I don't think would change their behavior, but I'd have to look into that. That's a good question.
Yeah, it just got me thinking because, right, they're ectothermic, and we know that when they have some kind of infection, viral, bacterial, sometimes they'll change their basking regimen to counteract that. Obviously, this isn't the same pathological, method, but I was just wondering if maybe that was.
Yeah, because I mean, with selenium, for instance, like Krista asked earlier, some lab studies have found that there's been sloughing of nails, kidney failure, and possibly even mortality with higher concentrations of selenium. But it's hard to compare those lab studies to in situ, like in the field. These turtles are in a wetland in a field, because they're not being fed 1,400 milligrams per kilogram every day for 7 days, So those different, I guess, regimens, if you will, like they're accumulating the metals in the environment different than they would be in the lab. So it's like, are these side effects actually happening in the field? I don't know. And if they probably happen over the long course of time, because of how they're accumulating and how quickly they are, I guess.
Gotcha. Thanks.
Okay, so I think that's it, Darien. So thank you, everybody, for attending. If you want to say anything to Darien, go ahead. We'll take about a 5-minute break, and then the committee will get back together, same location. So all right, thank you all.
Thanks, everybody, for coming. You guys are great.